Bacterial total protein and membrane protein extraction. Creative Biomart. This post describes several methods for extracting bacterial total protein and membrane proteins. 1. Reagents. Lysate, pH 8.5-9.0, 50 mm tris HCl, 2 mm eta, 100 mm NaCl, 0.5% Triton X100, and adjust pH to 8.5-9.0. Lysozyme. PMSF protease inhibitor. SDS 1%. Trizol lysis solution. Anhydrous ethanol. Isopropanol. 0.3 m guanidine hydrochloride. Hydrophobic protein extract, non-lysing solution, 1% Triton X114, 150 mm NaCl, 10 mm tris HCl, 1 mm eta, and pH adjusted to 8.0. 2. Equipment. High-speed centrifuge. Ultrasound system. Dialysis bags. EP tube. Vortex. Water bath and so on. 3. Procedure. 1. Extraction of total protein from fresh samples, simple method. 1. Self-made lysis solution, pH 8.5-9.0, 50 mm tris HCl, 2 mm eta, 100 mm NaCl, 0.5% Triton X100, pH adjusted to 8.5-9.0. Add 100 μg/milliliters lysozyme, 1 μl/milliliters protease inhibitor PMSF before use. 2. 40 milliliters of the bacterial solution was at 4 degrees Celsius for 15 minutes. The precipitate was washed twice with PBS and precipitated by adding 1 milliliter of the lysate to suspend the cells. 3. Ultrasonic crushing, using 300 watts. 10s ultrasound/10s interval, ultrasound 20 min, repeated freezing and thawing ultrasound 3 times to clear the liquid or discoloration. 4. 1000 grams centrifugal to remove large debris. The supernatant can be directly denatured after phage electrophoresis, or use 1% SDS solution after dialysis to freeze. Disadvantages. Western blotting results show that hydrophobic transmembrane protein extraction efficiency is limited. Isolate the total protein from trizol lysate. 1. After trizol dissolved sample broken, add chloroform layer, 10,000 grams centrifugation 15 min under 2-8 degrees Celsius, the upper aqueous phase for RNA extraction, and the volume is about 60% of the total volume. 2. Precipitate the DNA and the intermediate layer and the organic phase with ethanol. Add 0.3 milliliters of anhydrous ethanol each using 1 milliliter trizol. Room temperature for 3 min, 2-8 degrees Celsius, not more than 2,000 grams centrifugation for 5 min. 3. Move the supernatant to a new EP tube and precipitate the protein with isopropanol. Add 1 milliliter of trizol to 1.5 milliliters of isopropyl alcohol at room temperature for 10 min, 12,000 grams at 2-8 degrees Celsius for 10 min, and discard the supernatant. 4. Wash with 95% ethanol containing 0.3 m guanidine hydrochloride. 1 milliliter trizol, add 2 milliliters lotion, room temperature for 20 min, 2-8 degrees Celsius, 7,500 grams centrifugation 5 min, discard the supernatant. Wash 2 times. Finally add 2 milliliters of anhydrous ethanol, after vortexing, room temperature for 20 min, 2-8 degrees Celsius, 7,500 grams centrifugation for 5 min, discard the supernatant. 5. Freeze-dried 5-10 min, dissolve in 1% SDS solution, repeated blowing. 50 degrees Celsius temperature bath to completely dissolve, 2-8 degrees Celsius 10,000 grams centrifugation 10 min to remove insoluble Extraction of hydrophobic membrane proteins from fresh samples. Triton X114 detergent method. 1. Prepare hydrophobic protein extract, non-lysate. 1% Triton X114, 150 mm NaCl, 10 mm tris HCl, 1 mm ETA, pH adjusted to 8.0. 2. Bacteria were collected at 4 degrees Celsius after 15,000 grams centrifugation for 15 minutes. The cells were washed for 3 times with 1 milliliter of PBS containing 5 mm MgCl2, and finally 15,000 grams centrifugation at 4 degrees Celsius for 15 minutes. 3. Cell precipitation by adding 1 milliliter cold extract, placed at 4 degrees Celsius for 2H. 17,000 grams centrifugation 10 min, remove the precipitate and take the supernatant. 4. The Triton X114 content in the supernatant was increased to 2%, and 20 mm Kcal2 was added to inhibit partial protease activity and allowed to stand at 37 degrees Celsius for 10 minutes. The liquid phase and the decontamination phase were fully separated by centrifugation at 1000 grams for 10 minutes. 5. The liquid phase and the decontamination phase were separated, respectively, with 10 times the volume of cold acetone to precipitate on ice for 45 min. 6. Centrifuged at 17,000 grams for 30 min at 4 degrees Celsius and washed 3 times with deionized water. 7. The precipitate was dissolved in 1% SDS solution. Determine the protein concentration, and compare the extraction efficiency of the protein in the liquid phase and the decontamination phase. Generally, the hydrophobic membrane protein in the decontamination phase was more suitable for further protein experiment. 8. SDS page was used for further analysis of liquid and decontamination phase of the protein map. Thanks for watching. Contact Creative Biomart. Address: 45-1 Ramsey Road, Shirley, New York 11967, USA. Email: contact@creative-biomart.com. Website: http://www.creativebiomart.net